Hello, everyone. I'm Noor Jerboun. I graduated from Jordan University of Science and Technology, where I earned my master's in medical science with a major in genetics and molecular science. More recently, I received my doctorate from the University of Wollongong in medical neuroscience, specializing in the molecular and behavioral aspects of antenatal depression. I'm passionate about unrevealing the complexities of diseases to provide better cures. My professional journey began as a pathologist, and I later transitioned into research and lecturing in medical science. Today, I'm delighted to present my mini lecture on Ewing sarcoma disease, exploring its etiology and pathophysiology. So I will guide you through an introduction to what Ewing sarcoma is, followed by an exploration of its causes. We will delve into the progression of the disease, and for the sake of time, I will briefly highlight key aspects for identifying Ewing sarcoma, outline the current approaches to managing the disease, and then introduce some key elements aimed at enhancing future treatment approaches. Ewing sarcoma is a highly metastatic tumor originates from a transformation of mesenchymal stem cells that affect both bones and soft tissues. It's documented as the second most prevalent bone tumor with an incidence of nearly 3 patients per million every year. Patients with Ewing sarcoma typically present symptoms such as swelling in the affected bone, fever, fatigue, and weight loss. This cancer is known for its propensity to metastasize to the lungs, other bones, and the bone marrow. It has been observed to be more common in Caucasians, with a higher incidence in males than females, peaking in the second decade of life. What's the cause of sarcoma, of Ewing sarcoma? The exact cause of Ewing sarcoma is unknown, but it is thought to be as a result from a combination of genetic and environmental factors. Environmental factors may include exposure to radiation or certain chemicals. However, there is no strong evidence to support these links. Regarding genetic factors, Ewing sarcoma patients exhibit specific chromosomal translocation. It was found that 85 to 95% of these chromosomal translocations of Ewing sarcoma cases occur between the Q arms of the chromosome 22 and chromosome 11. This translocation fuses the Ewing sarcoma breakpoint region 1 gene on chromosome 22 to the friend of leukemia virus integration site 1 gene on chromosome 11, resulting in the EWS FLI1 fusion gene, which encodes for the EWS FLI1 fusion protein. Now, it was found that the formation of the EWS FLI1 fusion protein alone is not sufficient for the disease, as it was indicated by previous in vitro study. However, the fusion protein does influence key changes resulting in Ewing sarcoma. These key changes were reported to be 3 main changes. The first one, dysregulating gene expression by disturbing the signaling pathways which occur by binding the EWS FLI1 fusion protein to specific DNA sequences, influencing gene expression related to cell growth, differentiation, and survival, which leads to uncontrolled proliferation and tumor formation. The fusion protein was reported to inhibit programmed cell death, which known as apoptosis, enabling cancer cells to survive and multiply. Additionally, the EWS FLI1 fusion protein was found to induce angiogenesis, which is the formation of new blood vessels, supplying cancer cells with the necessary oxygen and nutrition for growth. Now, after the symptoms appeared, that there are 3 main diagnostic methods for the Ewing sarcoma. The first involves imaging, including X-ray, CT scan, and MRI scans. And as you can see here in these images, the tumor bone lump here is evident in both the arms and the pelvic images. The second diagnostic tool is biopsy. This process requires extracting tissue or cells sample of the affected area for examination to confirm the diagnosis and the cancer stage. The biopsy sample goes to microscopic test. As you can see here, the Ewing sarcoma cells appear as undifferentiated and characterized by a high nucleus to cytoplasm ratio. Then there is a cytogenetic test, which reveals the translocation karyotype, as illustrated here in this image of genome. There is a translocation between chromosome 22 and chromosome 11. The fusion part settles on chromosome 11, resulting in a deletion on chromosome 22. The third diagnostic tool is molecular tests that include fluorescence in situ hybridization, which enables visualizing of specific chromosome regions within cell nuclei using a fluorescent complementary probe DNA sequence of the gene of interest. The second molecular test is known to be reverse transcription polymerase chain reaction, or RT-PCR, which is achieved by utilizing reverse transcriptase to produce complementary DNA from the mRNA gene of interest, which is in this case the mRNA of the fusion protein, followed by amplification using PCR. The current treatment approaches of Ewing sarcoma are three primary approaches: chemotherapy, surgery, and radiation. With the available treatment options, the prognosis is 5-year survival rate for 70% of patients with localized Ewing sarcoma and 30% for patients with metastatic Ewing sarcoma. And at the end of my mini lecture, we emphasize the pressing need to enhance the cure for Ewing sarcoma. And as medical scientists, we recognize the critical importance of understanding the interaction between tumor cells and the microenvironment. This comprehension is vital for developing innovative cancer therapies that target not only the tumor cells, but also the surrounding environment. To achieve effective clinical therapeutic for Ewing sarcoma disease, we will still need a deeper understanding of the EWS-FLI1 fusion protein and its collaborating macromolecules and interactions. This may lead us to identify the Ewing sarcoma specific immune target cells to enhance the immunotherapy. Additionally, developing novel diagnostic tools for Ewing sarcoma, including discovery of novel biomarkers. For additional information, here are some valuable resources I utilized to prepare for this lecture. And of course, feel free to reach out to me via email if you have any questions. And thank you for your attention.